Hi everyone, welcome to our weekly AP Bio Chat. Today we're going to be doing a quick review of gene regulation, followed by a free response question sample, which we will go over together next week. We're pretty quickly approaching the first test date, and so if you have any questions, feel free to email me. My email address is here on the screen. You can also feel free to reach out to us on social media. For your reference, again, if you'd like to access any of our educational programming, we're doing a lot of different live online events. You can check out our calendar here. You can also reach out with us and engage with us on Facebook, Twitter, Instagram, or YouTube. This is just a reminder, and we're getting pretty close to the exam, so you probably don't need this reminder, hopefully. But we're going to be covering units 1 through 6 this year on the AP exam. Unit 7 and 8 will not be covered. And your exam date options are May 18th and June 3rd. You should reach out to your teacher or your testing coordinator to figure out which test date you're going to take your exam during. So gene regulation is basically the idea that within your genome, you don't necessarily want all of your genes to be transcribing mRNA and then therefore translating protein all the time, right? One, because this is wasteful. You don't necessarily need these proteins all the time. And so you want mechanisms that will allow you to selectively pick when you're going to transcribe and then therefore translate protein. So just a quick review. The process that we're talking about is part of the central dogma, right? So we have DNA, which is transcribed into RNA, and then RNA is translated into protein. This little schematic here is showing this happening in a eukaryote, but prokaryotes are also going to do transcription and translation. So gene regulation is going to be a topic that will apply to eukaryotes and prokaryotes alike. So that's important to consider. So there's going to be some terminology that I'm going to use that I want to make sure we're familiar with. So a regulatory gene is an important concept to understand. So I'm going to underline it here. A regulatory gene is a gene that synthesizes something called a regulatory protein. A regulatory protein is basically going to bind to the DNA at some location and then activate the transcription of a gene further down in the sequence, and therefore kind of acts like a molecular sort of light switch, right? All right, so this is the general overview of what we're talking about with gene regulation. It's the mechanism by which we can control which genes are being expressed. Let's clear this. So an example of where gene regulation can be observed is just by looking at phenotypes of organisms and of cells. So a big example here is that when we are embryonically developing, these embryonic, what we call stem cells, all kind of look the same, right? But based on which genes are turned on and off in subsequent generations of cells, results in what we call cell differentiation. In other words, different types of cells forming, right? So an example of this might be something that looks like this, right? So for example, with your blood cells might be expressing and turning on expression of a gene called hemoglobin, right? Your nerve cells probably are going to have that switch turned off. And then, your liver cells are going to be expressing enzymes that help you break down toxins in your body, right? So the basic idea here is that not all cells are going to need to express every single protein that your genome is capable of expressing. They're going to be expressing different genes based on what their function is in the body, right? And so gene regulation is observable just through diversity, right? Through diversity of cells, but also through diversity of different organisms. So we're going to talk a little bit about the difference between prokaryotes and eukaryotes. And the basic idea is that in prokaryotes, as is the case for most things in biology, the story is a lot simpler. So in prokaryotes, you actually have clusters of genes all in one place that are achieving the same sort of goal. And we'll look at two classic examples of that, the Lac Operon and the Trip Operon, which you've probably seen in class. In eukaryotes, because the genome of eukaryotes is generally much larger than in prokaryotes, we need a mechanism that allows us, and so therefore, genes that accomplish the same things are located maybe on different chromosomes or maybe even, thousands of base pairs apart. We need a mechanism that accounts for that just like greater space. Okay, so I'm going to go through the Lac and Trip Operon first, just because they're simpler and they're kind of nice, easy stories to latch onto. So the Lac Operon is named accordingly because it's got these genes. So if we sort of look at the schematic over here on the right, it's got these genes LAC-Z, LAC-Y, and LAC-A that ultimately produce enzymes that allow bacteria to break down lactose, which is a type of sugar. Why would they want to break down lactose? Well, they might want to break down lactose so that they can access glucose, which is a monomer of lactose. And why would a living thing want to access glucose? Well, glucose is going to allow a bacterial cell to produce ATP, right? So that's why this operon exists. Unfortunately for bacteria, they're kind of limited in terms of where they get their food, right? Whoever their host is, right? If their host is me, I'm not necessarily going to be providing it with direct sources of glucose. So if I drink a glass of milk, this little operon here will allow the bacterial cell basically to break down the milk sugar, which is available to them, right? So they can't be picky with their food. So that's why we might see this sort of Lac operon even existing. So the Lac operon is an inducible operon. What that basically means is that at its normal sort of state, it's usually off, right? So if we look at the left sort of schematic over here, right? Usually when there's no lactose present in the cell, so let's underline that here. When there's no lactose, right? What ends up happening is this thing called a repressor, which is shown here in red, is able to bind to this thing called an operator. The operator you can sort of think of as the switch. The switch is going to turn on the LAC-Z, LAC-Y, and LAC-A, which will then allow for the transcription of RNA, which will then allow for the translation of enzymes, right? So basically the idea is that without lactose present, the switch is turned off because this repressor binds to the operator and turns the switch off, right? Okay, so what happens if there is lactose present? Well, if there is lactose present, the bacteria is going to want to be able to break down that lactose, right? And so what's going to happen is the lactose will bind to the repressor. The repressor is just a protein. And what happens when things interact with proteins? Well, those proteins change shape. So what happens is this repressor basically changes shape so that it no longer fits into the operator, right? And now that this operator is sort of unbound, what can happen is RNA polymerase can bind to it and express LAC-Z, LAC-Y, and LAC-A. And then the result is that, this E. coli cell will be able to produce beta-galactosidase, permease, and transacetylase, all of which are going to help with the breakdown of lactose, okay? And so this is called an inducible operon, again, because usually this little system is turned off. And when we use an inducer like lactose, right, we can turn the switch on. Okay, so that's the Lac operon, which you've probably seen before. I'm going to show you an example now of kind of the opposite, right? So we're going to look at the Trip operon, which is called a repressible operon. And the reason it's repressible, repress means to stop, right? So a repressible operon is usually its base state is that it is turned on and transcription is happening, right? But there are molecules that we can activate in order to turn the switch off, right? So before we start, let's just talk about where this name comes from. Trip is coming from the amino acid name tryptophan, right? And our cells, and the same with bacterial cells, need tryptophan in order to build proteins, right? And so usually this trip operon consists of, instead of 3 genes here, the trip operon actually has 1, 2, 3, 4, 5 genes. And we've got the same sort of schematic where we have an operator that acts as the on or off switch. We've got the genes that we want to express or maybe not express, right? And so at its sort of base state, without the presence of anything, we would want to make tryptophan, right? Why? So that we can build protein. And so at its base state, the repressor, so we've got, the same sort of players here. The repressor is in red, and without tryptophan present, the repressor is actually inactive. So if you sort of look at the shape here, this can't bind. It can't bind to the operator, right? And since it can't bind to the operator, the effect is that this operator is in its on position. And so RNA polymerase is able to bind to the operon and transcribe the genes, and then therefore translate those RNA transcripts into the enzymes that will allow tryptophan synthesis to occur, right? But what happens if all of a sudden you find yourself as an E. coli cell in an environment where tryptophan is plentiful? If that happens, you probably don't want to waste your tryptophan making machinery because that requires energy, right? And so one way that we can respond to this is that you can sort of see here that the repressor will bind tryptophan when it's present, undergo a shape change, and that shape change will actually allow the repressor to be activated. And that activated repressor can now bind to the operator, blocking RNA polymerase from being able to access the rest of the operon, right? In other words, when tryptophan is present, the cell says, okay, I'm not going to be wasteful and make my own tryptophan when tryptophan is being provided to me, right? So does that sort of make sense from a cell energetics point of view? In other words, cells are smart, right? They're not going to waste energy synthesizing a compound that they already have access to. And so we've got this nice little feedback system here. And if we think about feedback, going back to our Lac Operon, right? Going back to our Lac Operon, we sort of see a feedback mechanism here too, right? Because if we are in the presence of lactose, what's going to happen? Well, we're making all of these enzymes that are going to break down the lactose, right? And once you've broken down all the lactose and turned it into glucose, which you can then use as a cell, well, then you're going to be back in this lactose absent sort of operon off state, right? If that sort of makes sense. So cells are highly efficient with these sorts of systems in place. All right. So eukaryotic gene regulation is a lot more complex. So unlike prokaryotes, we don't have operons, right? In other words, we don't have small enough genomes such that we can cluster all of our genes that do a specific job in one location. And so we need a system that allows us to turn on or off transcription of genes on a larger scale, right? And so instead of using a single sort of inducer system like prokaryotes do, we actually have a lot of proteins called transcription factors, which will bind to the DNA, create this sort of bend in the DNA, and create this loop complex that allows for the turning on or off of genes within a large segment of the genome. You don't have to know the details of this. You don't have to memorize or learn about the Tata box. Tata boxes, if you remember from your class, is just named after that TATA sequence in the gene that acts as sort of a promoter in the system. All you need to know is that the story here is more complex, and the reason for that is because of the larger genome size. We don't have operons, and so you need a system that will allow you to turn on or off genes that are located in vast regions of the genome at once. Another way that eukaryotic gene regulation is more complex than prokaryotes is that we also have something called histone modifications. So if you remember when you learned about the structure of DNA in cells earlier in the year, you probably learned that DNA is tightly coiled and forms these things called nucleosomes. And they actually have these proteins called histones bound to them, right? So what has actually been found to happen is that sometimes those histone tails can be acetylated, which just means an acetyl group is tagged onto the proteins. And what researchers have found is that when you add these acetyl groups to the histones, it actually loosens the chromatin structure, and therefore transcription can actually happen. So usually when you have these tight coils, you can't actually access the DNA and transcribe it. So let's like write that down. So tight coiling means that the DNA is inaccessible, right? But the cell has a way of making that DNA accessible again by adding acetyls. So when you acetylate, so let's say if we add acetylation, it loosens the coils, and therefore the DNA is accessible and transcribable. And so that's a way that we can sort of look at gene regulation in terms of, I guess, larger DNA structures. All right. I've been encouraging you to make sure you're familiar with keywords when you're studying. So I think the main keywords, I think a lot of times kids try and memorize like Lac Operon and Trip Operon. That's not necessary. I think it's more important for you to, be familiar with the idea of what an inducible region is, what a repressible region is, what an operon is, what transcription factors are, and so on and so forth. So this is the list of keywords I recommend you focusing on. And then we are going to wrap up the session by going through a quick sample free response question. So this is a sample DNA deletion experiment. A DNA deletion experiment is exactly what it sounds like. It's an experiment where you try cutting out certain genes or deleting them and seeing what the effect on the phenotype is, right? So let's go ahead and look at the graph and the associated text here. So it says the data was obtained from a DNA deletion experiment that tested possible control elements for the human gene MPGES1. This gene codes for an enzyme that synthesizes a type of prostaglandin, a chemical made during tissue inflammation. All right, so, as I'm sort of going through this as a student, I'm just going to go ahead and say that this is a study of gene regulation for a protein. Let's make this box bigger. That helps with inflammation. Oops. Let's see here. There we go. That looks better. And so it says the researchers focused on possible control elements in an enhancer region upstream of a reporter gene. So if we look at the diagram here, right, it looks like we're studying these possible control elements 1, 2, and 3, and it is upstream, meaning it's just located before in the sequence, this reporter gene. And the reporter gene, it looks like, is going to be measured. So it's the measured variable in terms of its mRNA transcript that is produced. So Swaraj, epigenetics is a growing field of genetics where we're sort of looking at environmental factors on our DNA. And the example of histone modification that I presented a little while ago is a big area of study for, so histone modification is an example of epigenetics. So it's sort of to answer your question, there are a lot of ways that epigenetics works. Histone modification is one. Okay, going back to this question here, the researchers focused on, so we're sort of looking at these different control elements We have this first sort of experimental group where none of them are deleted, and it looks like we have 100% of relative mRNA with respect to the control. And then it looks like we have 3 different samples where the first control element is knocked out, the second control element is knocked out, and then the third control element is knocked out. And if we sort of keep reading, they tell us to test the activity of each possible control element. They built 4 constructs, one with all 3 intact and one with each of the 3 deleted. Their results are summarized here. All right, so your job is this. Your job is to give yourself, I would say, so this would be an example of the first free response question that is 25 minutes long, and it's based on an experiment. So what I would do if I were you, I would pause this video right here. I'm going to get rid of my notes. I would try this on your own, and then what we'll do is next week we'll go over this one together. Make sure you're answering the questions. You can see that there's 4 different parts here. So you're determining the independent and dependent variable. You're labeling the construct that serves as the control group. You're explaining whether any of the possible control elements is an actual control element. And then they're asking you to propose a normal biological role for each control element based on the data. Really important here. So a distinction that I think could get confusing is that they're using, control group and then control element. The 2 are different. So control group is an experimental design thing, right? A control element would mean a piece of DNA that is controlling regulation of this reporter gene, right? All right. In any case, we will go over this next week. I would encourage you to join us then. So Thursday, May 7th at 2 p.m. And feel free to, in the time between now and then, suggest any review topics that you'd like to see us talk about or ask us any questions. You can feel free to email me. My email address is here, or you can feel free to message us on our Instagram page at DNA_learning_center. And then Swaraj, if we separate a population and spread different geographical area, can we find what are changed? Yes, absolutely. So there are definitely population studies with genetics and certainly for epigenetics as well, right? So I know that there are even, as we sort of as a global community undergo a really stressful time in our history, I'm sure there are epigenetic changes that are happening in our genomes right now. All right. Thanks for joining us, and we'll see you next week. Have a great day and take care.